wondered how scientists are able to research life-threatening genetic diseases like cancer, Huntington's, and cystic fibrosis? Well, an instrumental tool is at the foundation of this research, the DNA copy machine. In 1983, a man named Kary Mullis developed a technique that would amplify a piece of DNA in a short period of time. He named this technique PCR, or polymerase chain reaction. PCR is used in many fields of science, such as forensics and biomedical research. It was even used to sequence the genome of Steve Jobs' tumor. That's right, guys, Steve Jobs' tumor. Scientists used PCR to amplify his tumor DNA to be sequenced, which allowed him to receive personalized treatment based upon the molecular blueprints of his disease. This type of individualized treatment is called precision medicine and is at the forefront of patient care. In forensics, PCR is used to amplify a small sample of DNA, like hair collected at a crime scene, to be genetically fingerprinted and matched with the DNA fingerprint of a suspect. In research, PCR is used to edit genes to be studied, measure gene expression, clone pieces of DNA, identify specific genes in certain cells, and so much more. Now that we know what PCR does, let's jump into the actual reaction. Template DNA is the DNA you want to replicate. Its two strands are held together by hydrogen bonds formed by nucleotides, or the building blocks of DNA. These chemical letters pair up specifically with each other. A bonds with T, G bonds with C. They are also an ingredient of PCR and act as the ink in our DNA copy machine. This leads us into the first stage of PCR, denaturation. In this stage, heat is used to break apart the hydrogen bonds, which separates the two strands of the template DNA. The second stage of PCR is called annealing. This stage utilizes short, single-stranded DNA molecules called oligonucleotide primers. Annealing cools the reaction down so that the primers can bind to their complementary sequences on the newly single-stranded DNA molecules. The third stage of PCR is called extension. This stage features PCR's namesake and the star of the show, the DNA polymerase, an enzyme that synthesizes long chains of nucleic acids. In this stage, the polymerase binds to the 3' end of the primer and incorporates the nucleotide ink to synthesize new complementary strands of DNA. While these stages take place, a final secret ingredient called a buffer helps optimize the conditions for the reaction to occur. Most modern PCR methods take place in a thermal cycler, or a machine that heats the mixture at various different temperatures. This means that the DNA polymerase used cannot denature under high temperatures. A very commonly used type of DNA polymerase is Taq polymerase, an enzyme extracted from the thermophilic bacteria Thermus aquaticus found in the hot springs of Yellowstone National Park. The process is repeated 20 to 30 times, and each time the products of the reaction are used as the template DNA for the next, causing the amount of DNA to grow exponentially, hence the name chain reaction. And there you have it. My name is Annabelle Moth, and that was PCR, or the DNA copy.